A root tip is selected from those soaking in the holding solution. The root tip is placed into hydrochloric acid for a period of 4 minutes. So the root has spent 4 minutes in the acid. I'm now removing it and adding it to carnoids for 4 minutes to stop the effect of the acid. After 4 minutes, the root is removed from the carnoids and placed on a clean glass slide. The cells that are most actively dividing are only located at the very tip of the root, in this case about 2 millimeters. So that section is being cut and saved while the other piece is being discarded. The cells of onion have to be stained, since they themselves nor their chromosomes have any color. So here Mr. Hand is adding a drop of toluidine blue to the root tip. The root tip will soak in the stain for 2 minutes, just making sure the root tip is not floating on the stain, but is making good contact with it. So the root has been soaking in stain for 2 minutes, and now it's time to remove the stain using a paper towel. The paper towel is used as a blotter just to remove the excess stain from the slide. The idea is to remove the liquid stain, leaving the root intact on the slide. And that's good enough. Once all the stain is soaked up, a drop of water is added and a cover slip. Nice technique, Mr. Hand. As you can see, the weight of the cover slip alone was enough to crush the root right there. But in order to get a single layer of cells, a pencil eraser is used and downward pressure is exerted on the cover slip. And as you can see, the cells are becoming flattened. Just some paper towels to clean up the excess liquid. And we're ready for observation. And here's what this slide looks like. This is a scanner magnification. As you can see, the cells are very small. So we switched to 100x, and the cells are a little bigger, but still not large enough for you to discern the stages of mitosis. So we switched to high power, and at 440x you can see immediately a cell in anaphase at the pointer. Here's the cell at metaphase, chromosomes lined up along the equator. This one looks like it's an early anaphase. Chromosome pairs are just beginning to separate. Another metaphase cell. This cell looks to be in Late anaphase. I don't see any cell plate, so I know I'm not quite in telophase yet. Notice most of these cells are in interphase. I'm looking at the same slide in a different region, and I've come across a cell that looks to be in prophase. Nuclear envelope is still intact, however chromosomes are visible. Here's a cell that looks like it's an early metaphase. Here's a late anaphase. Notice how I'm moving in a zigzag fashion so as not to miss any cells. Another metaphase cell. Anaphase. I'm looking for the elusive telophase cell. I think I found one. Here's one. If you move the pointer slowly, you can see the cell plate. Move it to the, there it is, right there, cell plate.